I hope you can see my presentation. Yes, you can. First, also thanks to Svetlana for excellent introduction. Saved me several slides. So I'll actually, it was good that Svetlana was first. I'll build on what she presented and show some applications of the instrumentation and the methods we developed together and the application of these methods in the measurements that we have conducted over the last several years in China. Also to highlight our student Louise Brown, who Svetlana's and mine, who has been critically developing this instrumentation, as well as our colleagues from Hong Kong and Beijing, who have been supportive and enabled us to conduct these measurements. So as Svetlana said, the question is, what's the best measure for particle toxicity? So is it the physical properties such as number concentration, which is biased by nanoparticles, particle mass, or surface area, or is it the chemical composition? If it's the chemical composition, which one? And as she nicely pointed out, one way that we can sort of make a surrogate of total particle toxicity is to actually look and measure the oxidative stress or the concentration of reactive oxygen species. So either or of those. And that's all based upon that oxidative stress hypothesis, which says that PM introduces free radicals or produces free radicals in the cells, which cause oxidative stress, inflammation, cell injury, and death. So what's the best way to confirm this oxidative stress hypothesis or to be able to measure the toxicity of particles? There are several ways. We can have these sort of in vivo or human inhalation studies or animal inhalation studies, expose them to PM or human cell or cell cultures, and then look at the expression of oxidative stress markers, genes, etc. These are all very complicated. They involve biological specimens. The variation in between biological specimens and how they react is always a big unknown. And they are very expensive studies and need approvals, which is not always easy to obtain. To avoid all of that, we've developed these so-called cell-free or acellular assays to measure the concentration of ROS and radicals, or to measure the antioxidant depletion, as Svetlana mentioned, with those two different approaches, and then to be able, with both of those, to measure the oxidative stress potential. So what you actually need is you need some kind of cell-free or acellular assay, and we've used for years a cell-free acellular assay based on the perfluorescent nitroxide, which was developed at QET. So just in short, how these work. So these are, the nitroxide moiety is attached to a fluoropore in its normal sort of state. The fluoropore is not fluorescent. After reacting with a reactive oxygen species, there's a transfer of electron transfer between the nitroxide and the fluoropore, and the fluoropore either through reduction oxidation, and the fluoropore becomes fluorescent. So each reaction produces one fluorescent fluoropore, and measuring the changes in fluorescence, we estimate the concentration of ROS. So the first question we ask us are PM-bound ROS, and that's what these probes measures. Are they, with the Ristovski probe, are they a good marker for the health effects? And we've done a study in China with our colleague Lina Wang from Fudan University. Svetlana was also involved in this study, where we looked at the variability of cell upon exposure to cooking emissions, looking at different types of Chinese cooking emission. That was highlighted on the title page of ES&T when it came out in 2020. So, and then we looked at the cell viability versus the different physical parameters, let's say versus the total particle number concentration, and we see a fairly good correlation. So the smaller the concentration of particles number, the larger the cell viability. If we look at particle mass, we get a bit less, not as good correlation, not really clear as with particle number, but what really strikes out, if we plot the dependence of the total ROS concentration versus cell variability, we get this is measured by the PFN probes, we get a really good correlation. So this is really a clear indicator that what we measure with our fluorescent oxide probes really corresponds to the toxicity of the particles. We also have results we can find the paper showing concentration between ROS and oxidative stress markers with measure within the cell, so that what we measure really has some physical, chemical, has some meaning, toxicological meaning, and some toxicological value. So, okay, so we've got a probe which works well. So the next part is how do we measure that measure the concentration of ROS not only from sources, but be able to measure it over a longer period of time to find this sort of exposure or even to do the source apportionment of reactive oxygen species in urban environments. We need to develop a stationary continuous ROS monitor, and that monitor first has to sample the particles into a liquid. And once the particles into a liquid, they have to be mixed with your chemical probe, in our case, perfluorescent nitroxide. The perfluorescent nitroxide reacts with the ROS on the surface or in the particles themselves or in the captured PM. There's a change in the fluorescence, and from where we measure the ROS concentration. So that's done using an instrument called particle into nitroxide quencher, and this is the diagram of the or the photo sort of how the instrument looks like. It measures continuously or traps particle into a DMSO solution. So how it looks like, it has several parts. The first and the main part is the so-called particle into nitroxide quencher. So that is the part that actually captures the particle. So let's say the sample comes in, it's exposed to high concentration of water vapor. The sample is cooled down, the water vapor reaches supersaturation point. It condenses onto the existing particles. Particles grow to a certain size. They enter a cyclone, wetted wall cyclone, where the walls are continuously wetted with a DMSO solution containing our probe. The particles react with the probe. They are then taken into liquid to a debubbler to remove the bubbles through a fluorometer, and the change in the fluorescence is continuously measured. So what do we capture here? We actually capture both the reactive oxygen species that we have in the gas phase, as well as the reactive oxygen species in the particle phase. In order to just distinguish between the gas and the particle phase, we have to do two measurements. In the first measurement, we let the sample come, so both the gas and the particle phase, into the system, and we conduct one measurement getting gas and particle phase. Then in the second step, we put the sample through a HEPA filter, removing the particle phase, and then into the instrument. So we measure only the gas phase. The difference between the two signals that's shown here is the actual particle phase. So see, this is the fluorescence measurement. So 6,300 of some counts, fluorescent count. This is the measurement of the gas phase. And here, after a few minutes, we do the measurements of the particle phase. The difference between these two presents this is gas and particle, sorry, and this is only gas. So the difference is the particle phase. So we sort of continuously switch gas phase only, gas particle, gas phase, gas particle, and these increases or decreases are a measure of the particle concentration of the reactive oxygen species. And this is how the fluorescence of our signal looks during the measurements, the wavelength, and we have these two sort of peaks. That's the laser scattering of the laser light, and the fluorescence is somewhere around 485 nanometer first peak and 515 the second one. What are the properties? What are key PINC or our instrument, real-time instrument specification? It's capable of capturing any particles, trapping them which are larger than 20 nanometer with high efficiency. PM-bound ROS, we need around 2.5 minute time resolution. 40 seconds is the response time, so that's this time here needed for the signal to rise and to achieve a stable value. So on average, around 2.5 minutes is roughly the time resolution between the two measurements, which is fairly good. And it can work autonomously with around 30 minutes of daily maintenance. You need to replace the liquids that are fed into the instrument. And longest continuous operation was 5 weeks in Beijing, and I'll show 2 of those weeks in some later slides. To compare to other real-time instruments which measure similar, which are based on the DCFH assay. Why that one? Because that's commercially available, and I won't go to the name, these are just some of them. Just to note, they use different capturing methods, but all are more or less similar to this sort of type where you condense water onto particles, grow them sufficiently enough so you can capture them and then bring them back into a liquid where you have your probe. Flow rates are various from 5 to 16, 20 liters per minute, but you see the time resolution is larger than ours. Not that that's really critical if you're doing outdoor monitoring, but it's worth noting that we are several times faster, and our low detection limit is below 0.01, which means we have a significantly more higher sensitivity than the other methods. With the results I'll present are the 3 measurements that 3 measurement campaign that we did in China, in Hong Kong, Heshan, which is fairly close to Hong Kong, but on the Chinese side in the Guangdong province, and in Peking University. These were done over several years. Why China? Because obviously the pollution levels are much higher there. Our instrument struggles to measure anything in Brisbane air so far. We have to further improve the sensitivity of the instrument in order to be able to measure in cleaner environments. The main difference between the campaigns was that in Hong Kong, the instrument was manually operated, so one sample was taken every hour. So literally a person who was conducting one measurement every hour on hour for 24 hours over 2 weeks, I think, in September, October. So much smaller time resolution. Measurements were conducted at 2 sites, at Hong Kong City Uni rooftop, and at a background, so that was a background site, and at a roadside site, so very close to a road. And in Heshan is an industrial city in southern China. We had it automated at 5-minute sampling, October, November, for about 2 weeks, so highly polluted area in south of China. And that was October, November. And after that, the instrument went to Beijing, measured at Peking University in the College of Environmental Sciences, the 5-minute intervals during December month, so mainly in winter time. What are sort of the main highlights of the results from Hong Kong? These graphs shows we were looking at what's the best marker, or what is the oxidative potential or concentration of ROS best correlate with. So in Hong Kong, we saw there an interesting correlation between black carbon, which is a marker of traffic, and the oxidative potential. And we look at the measurements roadside and rooftop at weekdays and over the weekend. So interesting that we see that for high concentration, low concentration of black carbon, we see a correlation between the oxidative potential and the ROS, the oxidative potential and black carbon, but at high concentration, this sort of correlations falls apart. If you're at roadside measuring fresh, ROS particles that are emitted mainly several minutes, if that, before you capture it. If you go to the rooftop where particles needed a much longer time, so they were exposed to atmospheric aging, the correlation it sticks much better. So I think this is the role of the atmospheric aging processes. We also looked at the volatility, so-called, or the dependence of the ROS concentration on, are they tied to black carbon, or they're more tied to something that goes together with the black carbon, such as the volatile organic material that's emitted from traffic emissions. And one way to do that is to pass your sample to a so-called thermal denuder, in which you would expose the sample first to a elevated temperature of 50, 100, or 200 degrees. And in that process, you evaporate off the semi-volatile components. And we see that between ambient and just running it through that thermal denuder at room temperature, there isn't any difference between the total and the gas phase. And as we heat up the sample, so expose it to higher and higher temperatures, the ROS signal drops. And that's another confirmation that the semi-volatile particulate matter is actually responsible for the measured ROS signal that we had. So moving further on to measurements conducted at Hersham at the EPA station in 2018. So that's the instrument. It was on the rooftop. It was a school building below us. In addition to ROS measurements, PM2.5, PM10, ozone, NOx, sulfur dioxide, CO, temperature, humidity, and wind were also measured, and it operated for 3 weeks. So these are the results that we obtained. So the top graph shows the ROS concentration, PM2.5, ozone, and CO. And you can see these sort of daily peaks in ozone, so the photochemical occurrence of the peaks. We see an episode of very high pollution episode with the concentrations above 100 microgram per cubic meter, while other episodes where we had high ozone, we did not have such a high concentration of PM. So what we see is the average daily concentration was around 0.044 nanomoles per cubic meter over the whole measurement campaign. There was a strong diurnal variability. So we see increases of ROS clearly during most of the days. I'm not sure you can see that at the end as well. We also see correlation with ozone. So the increase of ozone leads to the increase of the ROS, but we also see correlation with CO. So if we look into a bit more into that, dig a bit deeper into that correlation between ROS, ozone, and CO, and we make a graph where we show the carbon monoxide CO concentration versus PM-bound ROS, and where we color the dots with the ozone concentration. And we see that we have a grouping at high ozone concentration and a grouping at low ozone concentration. So if we divide these into 3 groups, low ozone, less than 50 micron, 50 ppm roughly, between 50 and 100, and above 100 ppb, of ozone, we see that the higher the concentration of ozone, the larger the slope and the better the correlation. So the slope goes from 0.022 at low ozone concentration to 0.15, so almost a 10-fold, 7-fold increase in the slope. So dependence between CO and ozone. So what does this tell us? So higher ozone results in a higher slope, and that tells us if CO2 is an indicator of high organic PM concentration because it's a marker for traffic emission, ozone indicates high concentration of atmospheric oxidants. So if you have a combination of high CO, so high organics from traffic emission, in combination with high concentration of atmospheric oxidants, you get the formation of the so-called short-lived reactive oxygen species. So we see that there's a sort of the secondary form. So is this something new? No, it's not. We saw a very similar thing. This is something we've done with our colleagues from Cambridge, where we measured the concentration of ROS in a so-called flow tube where the secondary organic aerosols were produced by mixing a precursor gas with OH radicals on ozone and UV lamp and humidity. And secondary organic aerosols were formed, and the ROS were measured in this case with an impinger. And what we see is that the longer the residence time in the tube, so residence times up to 10 minutes, your concentration or intensity of the fluorescence divided by the total amount of SOA drops. So this is a measure of, let's say, concentration of ROS per milligram or per microgram of SOA produced. So for short residence time, you have a high concentration. For longer residence time, you have a smaller concentration. So this is a clear evidence of the existence of the short-lived reactive oxygen species that are produced in secondary organic processes. So if we apply this knowledge and we try to model the concentration of ROS, these are the x's, with short-lived and long-lived fractions of ROS, where the long-lived are proportional to your PM. So let's say they come from far away, they are there, and they live, they exist for a longer time, versus the short-lived ROS, which are proportional to CO and ozone. We see that we combine these two, that we can fairly well predict the ROS, total ROS concentration. So just by measuring the CO, ozone, and PM, we can, in Hersham, for a specific situation, predict the ROS concentration. And when we apply that same model and say, okay, let's look at 2 months of ozone, PM, and CO concentration at that site, and estimate what would be the contribution of short and long-lived species, we see that the short-lived species contribute around, where is that? This is the short-lived, yes, around 33% of the total ROS, while on a daily basis they can be even up to 70%. So the remainder is the long-lived ROS species. So the short-lived ROS species, where I say short-lived is literally 5 to 10 minutes, is the lifetime we estimated of these reactive oxygen species. They are significant contributors to PM-bound ROS in Hersham, in the southern China. And applying the sort of similar approach to measurements conducted in Beijing, we did measurements and measures the concentration of ROS, and this is the concentration nanomoles per meter cubic, normalized it by the particle mass, but we also measured the PM2.5 concentration, ozone, CO, and in this case, NO. And in Beijing, so this is the ROS concentration, we had 2 episodes of highly polluted episodes, so with high concentrations of PM going over a long period of time, and 2 clean episodes with fairly low concentration of PM. And what's interesting to note is in Beijing, it's much norther, it's 2,000 kilometers, I think, norther than Guangzhou, significantly different climate, so it was minus when we did these measurements, while in Guangzhou, the temperature didn't drop below 20 degrees or 25 degrees, more or less at the same time. But what's interesting to note is that during high pollution episodes in Beijing, the concentration of ozone is fairly small, while in the clean episodes, we have high concentration of ozone. Again, much smaller than you would get in Guangzhou. Guangzhou, the South China, has a much bigger problem with ozone, where the peaks are somewhere at around 100 ppb. So, and then if we plot what's the best correlation that we get, so what does our ROS correlate with? And we divide that, we look at the ROS concentration, so particle-bound ROS versus PM2.5, organic carbon, and this is elemental carbon or soot, and we look at total during high emission episodes, so polluted episodes, and during clean conditions. And what we see is that we get fairly good conditions, good correlations between ROS and all the measured parameters, but especially the organic carbon concentration. That's the best correlation, but during the low pollution episodes, these correlations are fairly small. And if we apply the same approach with short-lived and long-lived species and say, let's say that the concentration of long-lived species is proportional to your PM 2.5 concentration, and the concentration of short-lived species is proportional to CO and ozone product. Then, and we do the fitting or source apportionment, and this is your total PBRS concentration, and with the blue part is the stable or the long-lived RS, and the red part is, my Chinese colleague call this flexible, or I would call them short-lived RS. We can apportion the short and the long-lived. We see that during the polluted episode, the stable or the long-lived concentration of RS is significantly higher than the short-lived RS, while during the clean episodes, and this is an interesting one, the short-lived RS dominate because the concentration of PM to the RS that's brought to that air mass is fairly small, so your short-lived RS dominate over these periods. So in conclusion, we see that the average concentration in the southern region of China, where the UV is proportionally higher, is around 0.044 nanomoles per meter cubic, while in Beijing it is only 80 nanomoles per meter cubic. The main sources of pollution in Beijing, as we know, come from coal combustion, and during the winter period, the UV, or the amount of atmospheric oxygen, is fairly low, and the main contributors to the pollution are inorganic components, which do not contribute at least to particle-bound ROS. And assuming that they're short-lived, with the half-life of 5 to 10 minutes on average, they contribute to around 1/3 of the total PBRS in Hershan, but can be as high as 50% on days, again, which days, where the concentration of PM is small, but the ozone goes high, and similar observation in Beijing as well, when the concentration of PM is low, then the contribution of these short-lived reactive oxygen species becomes much more dominant. So why is this important? A lot of measurements, and even the ones that Svetlana showed, are done by collecting particles on filters or in different media, and then conducting the analysis significantly later, not in the matter of minutes, but sometimes even hours. And then you would question, if you have these short-living species, and they can be significant contributors, 30 to 50% on day-to-day basis, are these really methodologies properly done? And are we missing a really important component of the reactive oxygen species, the short-lived ones, and which can have significant health effects? So what we need to further do, we need to conduct more of these type of measurements, and we need to link the exposures of ROS, both short and long-lived, to epidemiological data. And I know that's a really big challenge, because most of these instruments are still in developmental phase, but hopefully in the near future we'll be building several and placing them for a longer time in China, and we'll be able to get some better data. And at the end, just to a few acknowledgements to all of my colleagues who have supported and helped in collecting this data. I did not put them all as co-authors, my postdocs and students as well, and also the funding bodies that contributed and funded that, the Australian Research Council, and the number of funding bodies both in China and Hong Kong, as well as the Australia-China Center for Applied Science and Management, which was key in setting all this collaboration up together. And I also thank the organizers for the invitation. So I hope I didn't go for way too long. Thank you.
Thank you, Zoran, very much for this presentation. I think both yours and Svetlana's presentations are extremely rich. I don't know if anybody has an overview of potential questions, but in the meantime, how many studies? So basically, are you the only group that is able to do this kind of studies where you look at the long term and short term? Because this could have a very big impact also when looking at sources in urban areas, how they develop. Yeah, how do you see that?
For the group from Cambridge University, Campbell's group, they use DCFH, which should be able, and Svetlana can correct me if I'm wrong, also to measure the short and long term ROS. The other sort of approach in these measurements, other group that has gone fairly far in these measurements, is the group of Vishal Verma from, is he from Chicago, from the States? But he uses a different probe, and he measures that oxidative potential more, not the concentration of short or long-lived species. So yes, there are other groups who can do that, and their results have slowly starting to come up from China. I think Campbell's group has done some measurements, and it would be worth actually approaching them and looking, can we reanalyze the data that they have collected using our approach with this short and long term. It's just a matter of analysis, so
yeah,
nothing else.
Well, is it a matter of is it a matter of analysis, or is it a matter of sample collection?
All three methods have this direct sampling, in the sense
right.
the sample is placed in water and analyzed immediately.
Okay.
In a matter of minutes.
Yeah, because I think that's I think is an extremely important point, that until it's solved, we won't be able to have more studies really. And I think the applications, of course, Svetlana had this list of open questions, and maybe there are some other ones that are actually towards sources and how with changed emission sources, how does the atmospheric composition actually change for the reactive species? And that will be, I think, very important as we keep changing the sources. Yes. So thank you very much for this. It was really extremely rich. I know you have been supporting Biopater for many years. And it's extremely interesting to see how the evidence is mounting and how the understanding and is mounting. So do you see any practical way how to connect this to epidemiological studies?
That's a fairly challenging thing. You really need long-term measurements, and these short measurements of species and that time resolution we are all using, they're very expensive. The amount of probes we chew through during the day is fairly large. So until we commercialize that and produce them in a more commercial, massive way, it's going to be challenging to conduct a study of 3 months, 6 months, a year. Really hard. Studies on analyzing filters for these sort of long-lived ROS, those are possible. And I think there's more and more of those, and they're slowly starting to come up. But that's, I think, only one side of the coin.
Yeah.
It really won't give the full picture.
And last question I have, which I'm not sure if it is really a good question or not. I know that people working with engineered nanoparticles, they do a lot of tests basically on cells. Is there any, I mean, obviously the natural or combustion nanoparticles, they are different, and they cannot be exactly treated the way, because you can't characterize them in the same way and so forth. They have a total different properties. And but the question, I think, is are those tests that are being used for the toxicological tests that are being used for the engineered nanoparticles, does it make any sense to try to apply them for example, the extracts from the combustion nanoparticles?
I don't know. What do you think, Svetlana? I'm not too supportive on that, but.
Could you repeat the question? So we extract extracts of combustion particles. What do we do with them?
Would it be worth conducting toxicological studies? I don't think so, to be honest.
Perhaps it's not a very good question, as I said.
No, you know what? These toxicological studies are being done constantly, and we are gaining proof for different particles. Like now, as I'm working more with engineers in any occupational settings, especially in production of new materials, they have to do this. And work safe procedures, like they have to have some measure of their exposure. Sorry, this is Sophia. So in some cases, serious organizations are conducting toxicological studies, and we were doing, like together, Zoran and me, we did a number of toxicological studies of combustion aerosols. We were exposing directly lung cells, and we were getting information. Sophia, please. Okay, Sophie's upside down. So it can be done. It is being done, and I would say we are documenting more and more information about different particles. In Finland, I was working with metal oxides. So yeah, it is being done. It's being done. And I would add to Zoran's discussion just that I do think, although limiting, the first and a very, very important step forward is that at least we do ROS source enforcement to filters. Because, you know, it is very unlikely that we will be able to supply EPA stations with this expensive piece of an equipment, but still they are collecting filters, and still every major EPA in capital cities, they are doing source enforcement. So if we get a quarter of that filter, and if we do OP measurements on that, we will generate maps, and we will have the source apportionment of ROS that can really help us improve epidemiological models. And this was something that Urs was talking about when he came down to Australia. He was talking that would be the step forward. That PM2.5 is still a bit outdated metric, especially that we are now producing materials and particles that are smaller and smaller. And they are more and more organic. So we will never capture the magnitude of their health effects just by measuring the mass. We would need to introduce a metric that we would agree on, that will add to our understanding what is dangerous, what's not.
Is anybody working with like sampling techniques? Because if you had the normal gravimetric sampling, and then if you were able to take additional samples, maybe on some other media or using some kind of other preservation techniques or something like that, is anybody investigating that, or is it not considered as a viable approach?
Well, I think Zoran would agree. The pink that was developed at QUT, it has the capacity. It can be made as a really small cyclone, and it can collect particles directly into aqueous medium. And that is going to alleviate like huge, gigantic mass of work. You know, people who ever analyze filters know how difficult it is to remove particles and to get a representative
artifacts that you get.
And artifacts are gigantic. You have evaporation from filters, condensation on filters. And, you know, once we got some sticky urban samples from Netherlands, it was impossible to remove them. Like it was very small, like maybe I managed to remove 10% of the mass. And who knows which components I managed to remove. So I guess that if we, and a cyclone investment it's not big. It is something, you know, it is very modest, and that could make a huge difference and help chemists analyze things much faster. They would just get samples and pass them through this process of chemical assays.
It sounds like a priority for kind of commercialization or something. I'm sure you are working on that. Well, do we have any other questions from the audience or publicum or from Belgrade directly? If not, then I would like really to thank you very much for your great overview and very rich presentations.